Namaste, myself Name-Shivi, Saxena, Faculty of Physiotherapy and Diagnostic. My discipline is Medical Laboratory Technology. My topic is Urease test and Acetate test, useful for BSc MLT second SEM and BN Bio second year students. Urease test, medium used for urease test. Any urea medium, agar, Christian urea agar or broth, Stewart's urea broth. Urease test medium are often a sole medium or a part of panel like motility indoor urease test. An indicator of urease test phenol red. Urease test principle. Urea may be diamide of acid. It's hydrolyzed with the discharge of ammonia and CO2. Many organisms, especially people who infect the tract, have an urease enzyme, which is in a position to separate urea within the presence of water to release ammonia and CO2. The ammonia combines with CO2 and water to make carbonate, which turns the medium alkaline, turning the indicator phenol red from its original saffron color to bright pink. Preparation of Christian's urea agar. Peptone 1 gram, dextrose 1 gram, sodium chloride 5 gram, potassium phosphate monobasic 2 gram, urea 20 gram, phenol red 0.012 gram, agar 15 to 20 gram. Dissolve the primary 6 ingredients in 100 ml of water and filter sterilize. Suspend the agar in 900 ml of water, boil to dissolve completely and autoclave at 121 degrees Celsius and 15 psi for quarter hours. Cool the agar to 50 to 55 degrees Celsius. Aspectically add 100 ml of filter sterilized urea based to the cooled agar solution and blend thoroughly. Distribute 4 to 5 ml per cell tube and slant the tube during cooling until solidified. Composition and preparation of Stewart's urea broth. Yeast extract 0.1 gram, potassium phosphate monobasic 9.1 gram, potassium phosphate dibasic 9.5 gram, urea 20 gram, phenol at 0.01 gram. Dissolve all ingredients in 1 liter of distilled water and filter sterilize. Distribute 3 ml of prepared broth per sterile tube. Note: prepared media will have a yellow-orange color once prepared. Do not reheat and medium as the urea will decompose. Prepared media can be stored in the refrigerator at 4 to 8 degrees Celsius until needed. Procedure for urease test. For questions, urea agar. Strike the whole slant surface with an important inoculum from an 18 to 24 hours pure culture. Incubate tubes with loose caps at 35 degrees Celsius. Observe the slant for a color change at 6 hours and 24 hours unless specified for extended incubation. For Stewart urea broth, inoculate the broth with an important inoculum broth and 18 to 24 hours pure culture. Shake the tube gently to suspend the bacteria. Incubate the tube with loose caps at 35 degrees Celsius. Observe the broth for a color change at 8 to 12 or 24 hours. Result and interpretation. Organisms that hydrolyze urea rapidly, Proteus species, Morganella morgani, and a few Providencia strains will produce strong positive reaction within 1 or 6 hours of incubation. Delay positive organism, example Klebsiella species and Enterovector species, will produce weak positive reaction within the slant in 6 hours of incubation, which can be intense during further incubation. The medium will remain a yellowish color if the organism is urease negative. Example E. coli. The routine diagnostic laboratories, the urease test results read within 24 hours. The organism produces urease enzyme. The color of the slant changes from light orange to magenta. If organisms do not produce urease, the agar slant and but remain light orange, medium retain original color. Diagnostic and utility of urease test. Urease tests help for the identification of Proteus species and the differentiate it from other non-lactose fermenting members of the Enterovectoriaceae family. Urease test is employed for the presumptive evidence of the presence of Helicobacter pylori in tissue biopsy material. This is often done by placing some of crushed tissue biopsy material directly into urease broth. A positive urease test is taken into account presence of Helicobacter pylori. Commercial available urease agar kits also are available. Rapid urease tests are often want to differentiate between the yeast Candida albicans and Cryptococcus neoforms. A preceptive identification of Cryptococcus neoforms could also be supported rapid urease production, whereas Candida albicans do not. Urea broth test, a standard non-invasive test to detect Helicobacter pylori, also supported urease activity. This is often sensitivity and specific test. Principle of urea broth test. Patient ingest radioactively labeled urea. If infection is present, the urease produced by Helicobacter pylori hydrolyzes the urea to form ammonia and labeled bicarbonate that is exhaled as CO2. The labeled CO2 is detected either by a counter a special spectrometer. Name of urease positive organism bacteria. Proteus species, Cryptococcus species, Conibacterium species, Helicobacter pylori, and Brucella species. Next test, citrate utilization test. This test is among a set of IMBIC tests, indoor methyl red, Vogus Boschke, and citrate, that are want to differentiate among the gram-negative bacilli within the enterobacteriaceae. Principle of citrate utilization test. Citrate agar is employed to check an organism's ability to utilize citrate as a source of energy. The medium contains citrate because the sole carbon source and ergonomic ammonia salt because the sole source of nitrogen. Bacteria which will grow on this medium produce an enzyme citrate priminase capable to converting citrate to pyruvate. Pyruvate can then enter the organism's metabolic cycle for the assembly of energy. Growth is indicative of utilization of citrate and intermediate metabolite within the Krebs cycle. When the bacteria metabolize citrate, the ammonium salts are weakened to ammonia, which increase alkalinity. The shift is pH turns the bromothymol blue indicator within the medium from green to blue above pH 7.6. Christians developed an alternate citrate test medium that does not require the organism to use citrate as a sole carbon sources. Christian's medium contain both peptone and cysteine. The citrate negative bacteria also can grow on this medium. A positive reaction shows that the organism can use citrate but not necessarily because the sole carbon sources. Media uses citrate utilization test. Simon citrate agar. Preparation. Dissolve above salt in deionized water. Adjust pH to 6 to 9. Add agar and bromomethyl blue. Gently heat with mixing to boiling until agar is dissolved. Dispense 4.0 to 5.0 ml into 16 mm tubes. Autoclave at 121 degree Celsius under 15 psi pressure for quarter hours. Cool is slanted position, long slant, shallow butt. Tubes should be stored during a refrigerator to make sure a time period of 6 to 8 weeks. The uninoculated medium are going to be a deep forest green thanks to the pH of the sample and therefore procedure of citrate utilization test. Stick the slant bag and forth with a lightweight inoculum picked from the middle of a well isolated colony. Incubate aerobically at 35 to 37 degrees Celsius for up to 4 to 7 days. Observe a color change from green to blue along the slant. Result interpretation of citrate utilization test. Positive reaction, growth with color change from green to intense blue along the slant. Examples Salmonella, Citrobacter, Klebsiella, Enterobacter, etc. Negative reaction, no growth and no color change, slant remain green. Example E. coli, Shigella, Morginella, Escherichia, etc. Quality control of citrate utilization test. Citrate positive, Klebsiella pneumoniae, ATCC 13883. Growth blue color. Citrate negative, E. coli, ATCC 25922. No growth or trace of growth. Limitations of citrate utilization test. Some organisms are capable of growth on acetate and do not produce a color change. Growth is taken into account a positive acetate utilization test even within the absence of a color change. Tests with equivocal result should be repeated. The reaction of the medium alone are not sufficient for identification of the species label. Thank you.